My name is Nicole Kleinstreuer, and I'm the acting director of NICEDM. I realized that I probably should have put a slide at the beginning to tell you what NICEDM is, so I'll just tell you verbally that we are the U.S. federal resource for alternatives to animal testing. So we're housed within the division of the National Toxicology Program at the National Institute for Environmental Health Sciences, but we provide support to a committee of 17 different U.S. federal agencies that either require or consider chemical safety testing data and are interested in moving away from outdated, low throughput, potentially uninformative animal-based testing methods and moving towards more rapid, more human relevant alternatives to animal testing. So we have a very broad program of work that covers pretty much every ISID you can think of in the alternative space. And usually what we do is really focused around toxicological testing and regulatory decision-making and coming up with alternatives to animal testing to support those processes, including MPS. So this is a little bit outside of our wheelhouse in terms of more of a biomedical research area and doing infectious disease research. But I think as every scientist in the world felt at the beginning of the pandemic, we wanted to do something to help. And so that was the motivation for starting this global working group. And I have to make a special mention of Anthony Holmes, who's a senior scientist at the NC3Rs, which is the UK 3Rs organization, because he co-chairs this working group with me. So the founding members of this working group, as I mentioned, Anthony Holmes is the co-chair, and then we have representation from NCATS, obviously due to their leadership in the tissue chip space and the tissue chip consortium, and then from NIAID, which is the Allergy and Infectious Disease Institute in NIH, we have Mark Williams, and then we have two representatives from the Department of Defense, Tyler and Kyle, who are doing really excellent work. And you may have seen some of Tyler's posters at the poster session last night. And they're leading a lot of work in the tissue chip space, particularly as it relates to infectious disease research. So then we started reaching out to the community through the networks of our founding members. And one of our key objectives was to involve as many regulators as possible and folks in government agencies so that we could really start to have these conversations about what are sort of the readiness criteria, what are the contexts of use, what is going to help move MPS into the forefront as far as being acceptable for forming part of regulatory submission packages. And obviously we see COVID, as a disaster, but also an opportunity, partially because the animal models for COVID are quite bad. They don't get sick the way humans do. In fact, basically the closest one is the golden hamster. I don't know what that says about us as a species, but particularly the non-human primate models are very bad. And so there's this really strong opportunity for tissue chips to kind of step up and provide human relevant insight into the mechanisms of the disease, the pathophysiology, and maybe provide platforms for testing new therapeutics. So we have representatives from divisions, branches, or centers of all of these organizations. We have, I think, every biotech company that was here at the World Summit, or almost everyone represented on the working group. And again, if you're from a biotech company and you don't see your symbol represented on the slide here, we should remedy that. That's actually one of the objectives of the presentation today, is to continue to spread the word about the working group and recruit additional members. And then we have obviously some large pharma companies represented. These are the folks who are really leading the field in developing therapeutics, and so are quite interested in identifying relevant testing platforms that they could use to test their lead compounds. And then by far the largest representation that we have is from academics. So I think that's where, as we've seen over the last several days, so much exciting work is happening in the research space in developing new MPS models and platforms. And so we have so many different universities and groups from universities. This is actually just the first slide, and here's the second slide of all of the academic proteomics and MPS networks that we have represented on the working group. So really, what are the detailed objectives of MPSCoRe? It's to create this shared space. So to have this forum that is intended to be, a very neutral forum, but it wants to kind of facilitate this engagement and this interaction among international collaborators. We do have very heavy representation from the US, of course, but we have many, many groups in Europe, in Asia, in South America, elsewhere in North America that are part of the global working group, and we're always looking for more members. And we're really trying to facilitate connections to help researchers troubleshoot, help them talk and learn from each other's experiences and share resources and share advice. To really try and support the application of these novel technologies. And as I already mentioned, we are really engaging with regulatory authorities from around the world to try and understand what are the regulatory needs and decision contexts, and how can we help move the field forward in terms of the assessment of these novel systems and characterizing their performance and their readiness criteria. And then, of course, because my organization and NC3Rs are really leaders in the alternative space, obviously the 3Rs opportunities to reduce, refine, or replace animal testing are fundamental to this effort as well. There have been countless publications that have come out over the last several years on COVID disease mechanisms, many of which are being done in microphysiological systems. And actually, one of the resources that we have on our website is a survey of the literature on MPS and COVID. It's current as of the end of last year. And at the end of the presentation, I'll show you a link to where you can access that. So there's absolutely no way that I could possibly highlight all of the publications that have come out of members of the working group. So I've just chosen to focus on 2 publications. One of them is actually an overview that myself and Anthony wrote that was a feature article in Drug Discovery Today that came out at the end of last year, that described the working group, but also gave the results of a survey that we did of our membership to understand what are the challenges that MPS developers and researchers are facing. And so these range from scientific challenges, such as access to well-characterized cell sources, incorporating immune components, standards and performance criteria, access to tissues, and then also practical challenges. So things like resources and access to BSL-3 facilities, et cetera. And so we've had a number of very productive discussions in the working group on how to address some of these challenges now that they've been identified. The other publication that I'd like to highlight is from some members of the working group who also happen to be some of the organizers of the MPS World Summit. Then we partnered with them and researchers from NIAID to highlight really the critical necessity of pivoting the work on COVID-19 mechanisms and understanding of the mechanisms to a reliance on using microphysiological systems. And here, this was illustrated for the brain. So using the brain organoid models that have been developed at Johns Hopkins at the Center for Alternatives to Animal Testing, they were the first group to show that there is infection of COVID-19 in the brain. And this paper includes, again, a survey of the literature and the major findings, and then also all of the neurological symptoms that have been observed across many large cohorts of COVID patients, and then also postulates sort of the mechanisms of neuro effects from COVID-19 infection, which include things like cytokine storm and systemic homeostasis imbalance and hypercoagulation, ischemia, leading to many of these outcomes that you see on the right-hand side that range from actual physiological to neurological and psychological outcomes. So a couple of ongoing projects that are actually funded under the MPS for COVID Research Working Group are work that's being done at NIAID. So historically, the Integrated Research Facility, or the IRF at NIAID, has been very much an animal shop. So they have a very strong non-human primate program and many other animal models. And in part through discussions with the MPSCoRe working group, their leadership has really pivoted a little bit and expanded their program to now include MPS models. So they have brought some of the emulate systems in-house into their BSL-3 and BSL-4 facilities. And then the other project that's funded under MPSCoRe is to add some supplementary funding to the funding that NCATS has largely provided for development of the MPSDB, which now has a new name. And I will talk about both of these in just a little bit more detail. So the NIAID IRF program is also being done in partnership with the DoD. So Kyle and Tyler's group are providing lots of advice and training. And we are paying for the chips and helping with study design. And basically, this is looking at some novel therapeutics, including synthetically derived antibodies, testing those in MPS models. So starting with the lung chip model and comparing that with preclinical data and clinical data when it's available. And so they're using the golden hamster model, as I mentioned, and then they'll also be comparing with human data from clinical trials. So these are antibodies that are already approved and gone into clinical trials in Europe. And so it's a really, really nice opportunity to have, kind of a cross system comparison and stepping up in complexity from just your typical in vitro kind of vero cell based model to the MPS, to the animal model, to the humans, and seeing sort of what is the value of information that's provided by the MPS. And then, as I mentioned, the MPS database is now called the Biostatistics Analytic Platform, and hopefully you visited the booth or saw one of the excellent talks that referenced this. This is a really phenomenal resource for the MPS community. So you have the opportunity here to upload your data. There's varying levels of security, so you can choose to have the information that you upload be completely confidential and secure only to yourself or to your group or to a group of external collaborators that you define, or eventually you can make it open and publicly accessible. So that's a really nice feature. And then one of the other really nice features is all of the different tools and workflows that they have as part of the system. So you can do things like reproducibility analyses, you can compare across similar experiments that are targeting the same biology. You can look at ADME, you can do PBPK modeling. So it's really fantastic. And if you haven't checked this out, I highly encourage you to do so. What we specifically supported is the development of a COVID-19 disease model portal. So this includes lots of information on disease biology, different experimental models, direct links to vendors for specific resources, that can facilitate COVID-19 research in MPS. And it's really designed to be this sort of central repository for COVID-19 experimental data, not just confined to that coming out of MPS models, but also from preclinical and clinical work to try and, again, facilitate those comparisons. And there is a private portal for MPSCoRe members. So if you are a member of the working group, then you can upload your profile and upload information about your expertise and your platforms and your interests and availability of things like BSL 4 and 3 facilities. And then that is, again, designed to encourage interaction and connection and networking among the membership. Something else that we've just added some additional funding for is the development of an IPSC portal. So obviously IPSCs are a tremendous resource in this field, and there's really a need to try and find, so to have a repository or a resource where researchers can go and find information on iPSC models that are available and what is their maturation status, their mutation status, how to obtain them, different protocols, et cetera. And so at the moment, the information that's included in the iPSC portal is shown on the screen here. So it includes things like differentiation characteristics, patient profiles, what is the source of the cells, what are the protocols, and are there any established applications either in the healthy or the disease model spaces. And then we have a number of resources available online. So we had last year, as a satellite to the NCATS Tissue Chip Consortium meeting, we had a virtual workshop, and it was really a fantastic meeting. We had presentations from a number of our membership on the research that's ongoing in their groups, and that was recorded and is publicly available on our website. We also have webinars that are not recorded and are not publicly available that are intended as a forum to share unpublished or ongoing research information and ask questions and help troubleshoot among our membership. And so we have those every few months. Obviously, one of the resources is simply having those points of contact. So we have distribution lists. My group is always happy to provide study design support. We have many experts in validation of alternatives as part of the group, not only in our organization, but in organizations like CAT. And then, we also try and provide information on funding mechanisms wherever those are available, and have dialogue with regulatory decision makers to understand their decision frameworks and their contexts of use. And one really cool thing that we started doing at the end of last year was, in response to the emergence of variants of concern, we have a direct line to the WHO. So Simon Funnell is part of our group, and we call emergency meetings of the MPSCoRe work group when there is a new variant of concern, and then we can identify people that have models set up in their lab ready to go, and they can receive the variant from the WHO distribution centers and start testing them and start trying to understand what are the potential evolutions of symptoms and presentations of the disease that are emerging with these different variants. And then I already mentioned the Biocystic resource, and they have just a really fantastic series of training videos that's available online. So I'll stop there. I think I'm right on time. And we have about 130 members at this stage, and we're always welcoming new members. So please let me know if you'd like to join. You can also just go to our website there and find out more information. See we have the downloadable Excel sheet that has a survey of the MPS and COVID literature, current as of the end of last year, and we're updating that every few months. And that's also where you can find things like the recordings of the workshop. So thank you very much.